Yes, hello. So we are going to revise together paper 1 970011 October November 2020. Why I chose this paper is because it is having some interesting and tricky questions, as we shall be seeing. We can go to the first question. So the first question in this paper was which row shows the correct order of sizes of these cell structures? So there is mitochondrion, there is a ribosome, there is a cell surface membrane, and a chloroplast. So from our theory, we know that from our chapter 1, you know that chloroplast is by far the largest. So in the ranking, chloroplast will be number 1, and then number 2 will be the mitochondrion, and then this is followed by the ribosome, and then so this will be number 3. Then number 4 will be cell surface membrane, which is about 7 nanometers. So if we check in the options A and B, they can be eliminated on the fact that they are using one mitochondrion as the largest, and we know that is not true. Then we go for C and D. 4 is a chloroplast, so 4 qualifies. So we can agree that C and D, they are okay in terms of the first one. And then we go to the second option, 1 and 2. From our ranking, the second largest is a mitochondrion, so C is okay. But then D is saying the second largest is ribosome, so C is the most correct answer here. So we go with C, and D is eliminated. So moving on to number 2, it's about the eyepiece graticule and stage micrometer. So we know the largest, the biggest is the stage micrometer, and then the smallest for this case will be our eyepiece graticule. The question is, the stage micrometer scale is replaced by a slide of a plant cell. So this is a plant cell. What is the actual length of the nucleus in the plant cell? So when we come back here, they're telling us that the small divisions of the stage micrometer scale are 0.1 millimeter. So it means that moving from here to here, let's say from point A to point B, this distance is equivalent to 0.1 millimeters. So how many EPGs are those? It will be 50 minus 10, which will be 40. So it implies that 40 is equivalent to 0.1 millimeters on the stage micrometer. Now these are 40 EPGs. Now how about 1 EPG will be equivalent to 0.1 out of 40, but this is in millimeters. Okay, now the question is, the stage micrometer scale is replaced by a slide of a plant cell, and then they want us to determine the length of a nucleus. I think you need to be careful. It's not the whole cell, but rather the nucleus, and the nucleus should be this other bigger structure. So if we are to extrapolate around here, of course you have to use a ruler. So this is having about 10 EPGs. So if 1 EPG is equal to 0.1 out of 40 millimeters, then how about 10 EPGs? So it will be 0.1 out of 40 times 10, and this will be giving us 1 out of 40 millimeters, and this is equivalent to 0. I believe this is equivalent to 0.025 millimeters. Now if we are to convert this into micrometers, it will be 0.025 times 1,000, and this is 25 micrometers. So the answer becomes B, and most students went for C, whereby they measured the whole cell, and that was wrong. So moving forward to number 3, they are saying insulin is a hormone synthesized from 2 polypeptide chains inside cells of the pancreas, which row shows the structures that are used in producing insulin and releasing it from the cells. So there's Golgi body, rhizosome, and rough endoplasmic reticulum. So we know the protein synthesis happens around the rough endoplasmic reticulum, in particular within the ribosome, so this should be correct. And then Golgi body we know is also essential for the modification of these proteins. Then lysosomes are important in keeping the hydrolytic enzymes, but now for this case, to produce insulin we don't need lysosomes. So A, B, D, they are wrong. So we can go for option C, which is giving Golgi body and rough endoplasmic reticulum at the same time eliminating lysosomes. Now, number 4, the question was, the electron micrograph shows a cell structure in a cell, in an eukaryotic cell, So we can look at these structures within a cell, and then they are saying which statements about this cell structure are correct. You can see that this is a cross section of a centriole. So what is a centriole for? Is it for ATP synthesis? Automatically you know this is for mitochondrion. The cell structure is made up of protein molecules. That is true because it is made up of the tubular dimers. The cell structure replicates during interphase of the cell cycle. This is also true. These are all features of the centriole. So the answer would be 2 and 3. Number 5. 4 students were asked to match the function with appearance of some cell structures in an animal cell. The functions were listed by number. So these are the functions: synthesis of prepeptides. We know this happens within the ribosomes. Number 2, synthesis of lipids. This could be the smooth endoplasmic reticulum. And then packaging of hydrolytic enzymes that will remain in the cell. This could be our Golgi body or Golgi apparatus. The appearances were listed by letter. Membranes which surround an enclosed inner cavity. This could be smooth endoplasmic reticulum. Non-membrane bound spherical structures. This could be the ribosomes. A double membrane interspaced with nuclear pores. I believe this is a nucleus. Non-membrane bound cylindrical structures. These could be centrioles. Membrane bound sacs arranged as flattened stack. This is a Golgi body. So they are saying which student correctly matched the numbered function with the appearance of the cell structure. We can look at A, B, C, D. Okay, at A they are saying 1 for W, 1 synthesis of prepeptides and ribosomes. So W, non-membrane bound spherical structure, that makes sense. Then 2 for synthesis of lipids. Smooth endoplasmic reticulum. V. A makes more sense. When we check on B, there is Z. Z, which is membrane bound sacs arranged as flattened stack. They don't match, so we can eliminate B. And then C and D can also be eliminated given that they are saying 1 synthesis of prepeptides with Z, that is Golgi body. They surely don't match, so we can eliminate C and D, and then we are left with the best option as A, if you are to go through it critically. And then number 6, moving on to number 6, the DNA of typical prokaryotes is naked and circular. Which statement describes how the DNA of eukaryotes differs from the DNA of a typical prokaryote? A. Only DNA of eukaryotes has a nuclear envelope around it and is double helix. So first of all, it is true that it is having a nuclear envelope, and then being double helix. Also prokaryotes, they have got double helix. Then part B, they are saying only DNA of eukaryotes has a nuclear envelope. Okay, again the same thing, around it and is circular. This is not true. It's only for the prokaryotes that are circular. Then C, only DNA of eukaryotes has proteins attached to it and is double helix. Again, double helix is also for prokaryotes, even though it is true that the eukaryotes have got DNA proteins. Then lastly, D, only DNA of eukaryotes has proteins attached to it and is linear. So yes, we have proteins, those histone proteins, which are exclusively found in eukaryotes, not in prokaryotes. And again, it is true, it is linear, whereas for the prokaryotes, it's circular. So here the answer was D. Moving forward to number 7, the question is, a student carried out the Benedict's test on the two different samples of milk X and Y. A sample of each type of milk was heated to 100 degrees Celsius in water bath with Benedict's solution, and the time taken for the first appearance of a color change was recorded. So we have X and Y, and then the time taken for the appearance of any color change. So we can assume that the less the time, then the more the concentration of the reducing sugar. So for this case, X is having more reducing sugar than Y, but we can see that all of them had a reducing sugar. Which row shows the biological molecule the student detected in each sample of milk and the sample of milk with the highest concentration of the biological molecule. So the biological molecule present in each milk, glucose, it can be true. However, we know that there are also other reducing sugars, uh, which could be fructose, maltose. Uh, so all those are reducing sugars which can be found also in milk, lactose. That is A and B. Okay, it is more specific by saying glucose. So we can first leave A and B, and then when we go to C, they are saying reducing sugar, C and D. So this one leaves a room of doubt because it can be glucose, it can be maltose, or any other reducing sugar. So we can think about C and D. And then sample of milk with the highest concentration of the biological molecule, X or Y. So we can see that X is having the highest because it took less time. So the best option here was C, even though A would make sense, but we are saying the reducing sugar could be anything else apart from glucose. So the best option was C. Moving on to number 8, the diagram shows the structure of a monomer. So we can see this is glucose, but to be more specific, due to the position of this hydroxyl group, this is alpha glucose, and we know that alpha glucose can be a monomer of amylopectin. That is true. It can also be a monomer in glycogen. We know amylopectin and glycogen, they are actually almost the same, only that in glycogen we have more branching. And then sucrose, yes, sucrose is also made of alpha glucose and beta fructose. So the best option here was B. It can be a monomer for all of them. Now moving on to number 9, which molecules are formed by the condensation and then involving a carboxyl group. So amylopectin, collagen, triglyceride, sucrose. It is true all of them, they involve condensation, but for sucrose we don't involve the carboxyl group in the formation of those glycosylic bonds. So is amylopectin. So we can eliminate all these which are having amylopectin because these carbohydrates, we don't involve the carboxyl group in the formation of the glycosylic bond. So we are left with collagen, which is a protein, and also beta globin is also a protein. And we know to form the peptide bond, we need the carboxyl group and the amino group. And again, to form the ester bonds in the triglycerides, we also need the carboxyl group that reacts with the hydroxyl group in the glycerol. So here the best answer was D. Number 10 was which row correctly matches the function with the structural features of cellulose, collagen, glycogen, or triglyceride. So the molecule and then also the function plus the structure. Part A, they are saying cellulose. function for support, that is true. Being fibrous, it is true. Molecules held together by hydrogen bonds, they are involved, yes. Branched, no, it's not branched. Triglyceride, energy source, yes. And then fibrous, no. Molecules held together by hydrogen bonds, not really. Then branched chains, no. So here the best answer, so far it is A. Maybe we can check on other options quickly. For part B, collagen strengthening, that is true. Fibrous, true. Molecules held by hydrogen bonds, yes, they are involved. Branched chains, no. Then for cellulose support, being fibrous, okay. So we know that cellulose is not branched, it is linear, made of those monomers of beta glucose. So B is out. And then when we go to C, collagen strengthening, it is true. Fibrous, it is true. Hydrogen bonds, true. And then branched, no, collagen is not branched. Then D, glycogen for storage, yes, in animals. Fibrous, no, yeah, that is true. Hydrogen bonds, they are involved somehow. And then branched chains, yes. Then for triglycerides, energy source, yes. Fibrous, not really. Then held together by hydrogen bonds, no, it is the ester bonds which are involved in the triglycerides. So here the best answer was A. Moving forward to number 11, the diagram shows the amino acids in a polypeptide. So these are the amino acids like glutamine, histidine, valine, phenylalanine, etc. And then they are giving you a key saying that those which are polar, I think these which are shade, these which are those which are polar, this is a key, polar, and then also those which are non-polar, they are basically circular. So they could be polar, basic, polar, acidic, or polar, just polar. And then we also have non-polar, which are just circular. Anyway, so going to the question, they were saying, an enzyme catalyzes the hydrolysis of any peptide bond between a non-polar and any other amino acid. How many small peptides and single amino acids will be formed by the action of the enzyme? So they are saying that this enzyme is able to break bonds between non-polar and anything else that is surrounding. No, this is not true. So it will be the first cut will be between histidine and valine. Another cut will be between phenylalanine and threonine, THR, because it is between non-polar and polar. Another cut will be around this here, asparagine and valine. Then another cut will be between lysine and proline. Then we have another non-polar here. This is leucine and asparagine, so another cut here. Then where are we having another cut? I think between this methionine and glutamine around here. Where else we have this adenine and serine, again adenine and serine. Where else? Aspartate and methionine, then histidine and methionine, again another cut. Glutamate and phenylalanine, then phenylalanine and lysine, lysine and adenine and threonine. So I think those are the cuts we are having. So they are saying small peptides and then the single amino acids. I mean, from this question, they want us to count the single amino acids and then also the small peptides. So we can start with the single amino acids. So where are we having only one? I think this will be one. And then where else are we having another one? Moving forward, there's another one here. This will be the second single amino acid. Then we shall be having the third one here. amino acid that is just single. Then again, you can see also this lysine will be a single one, because so these are 4. And then they want us also to count the small polypeptides. So those are small chains of amino acids. So we have 1, 2, 3, 4. Then this is 5, 6, 7, 8, 9, 10, 11, and then 11, this will be 12, and then 13 will be here. So we have 13 small peptides, and then also we have 4 single amino acids. So here the best option will be D. Moving on to number 12, the question was, which properties of water reduce temperature changes inside the cell? Cohesion, not really, this is for the capillary pool. And then latent heat of vaporization, this is for cooling. Specific heat capacity, I think this makes more sense. We know that it takes a lot of heat to change the temperature of the water, so the best option here will be D. Okay, so moving to number 13. Number 13, the question was, which enzyme is extracellular? So these are enzymes which work outside the cell. So amylase in saliva is an enzyme that catalyzes the breakdown of starch in the mouth. Yes, I believe this is an extracellular enzyme. And maybe we can look at the different options which are there. ATP synthetase is an enzyme found in mitochondria that synthesizes ATP. This is found in the membrane of the mitochondria, so that is intracellular, it is working inside the cell. Then C, DNA polymerase, an enzyme that helps build DNA molecules by assembling nucleotides. This is true, but it's also happening within the cell. Then D, RNA polymerase, an enzyme involved in the process of gene transcription to form messenger RNA. It is so true, but again, it is happening within the cell. So all these three are intracellular, apart from D, the amylase, which acts within the mouth. Okay. And then now moving to number 14. Number 14 was a tricky question. The question was, the graph shows how the concentration of components of an enzyme catalyzed reaction changes with time. Which line represents enzymes with the empty active site? So I think the trick was here. Which line represents enzymes with empty active sites? So the concentration and then time. So we can assume that because we are monitoring the progress of a given reaction, at first we shall be having very high active sites which are free, but then as the reaction progresses, these active sites will be filled up by the substrate. But then what we expect, when the reaction nears the completion, again the active sites will be free, because we know enzymes, they catalyze the reactions, but then at the end of a reaction they are free. So we expect something like this. When we have to check on the options, for A they are saying the active sites will keep on increasing, not really. And then for B, we can see at start we had relatively high number of active sites, then they reduced because now the reaction is taking place, they are occupied. Then when the reaction is complete, the active sites are free again. So I think this is matching a little bit to what we are expecting here. And then C, they are saying the active sites will continue reducing forever. And then D, they increase with time now. So here the best option was B. Then number 15, which effects can non-competitive inhibitors have on enzyme control reactions? Lower the Km. So for non-competitive, we need to know its effect on Vmax. The Vmax is lowered, but then the Km, this is lowered. But then we need to remember that the Km remains constant. So that's the difference. Then on the this is non-competitive, and then on the other hand, for competitive, we know that Vmax stays the same, and then on the other hand, Km is increasing, and once it is increased, also the affinity of this enzyme towards the substrate is lowered. Anyway, so return to the question. They were saying which effects can non-competitive inhibitor have on enzyme controlled reactions? Lower the Km, so this is not true. The Km remains constant. Reduce the concentration of the product, obviously, because we're having an inhibitor, so the product formation will be reduced. Reduce the rate of metabolic pathway. Yes, the rate will also be reduced. So here the best option was D. Number 16, which process can allow transport into or out of the cell? Active transport, yes. Facilitated diffusion, yes. Osmosis, yes. So number 16, the best option was A. All of them can be involved. Moving forward to number 17, plant cells were put into one of the three different concentrations of sugar, 10, 5, and then 2.5. The cells were left 50 minutes and then observed using a light microscope. So when we see these shapes, we can see X a little bit normal, maybe it was in isotonic solution, and then Y appears to be tagged, and Z appears to be plasmorized. So they are saying which statements are correct. Number one, cell Y, it is this one. Cell Y had a lower water potential than the sugar solution it was put into. Let's think about that. Cell Y had a lower water potential than sugar solution it was put into. Yes, I believe so, because that's why it is gaining water, because it had lower water potential. So water moved from the sugar solution to the cell, and that's how it became tagged. Then number two, cell Z was put into 10% sugar solution, because when we look here, 10% is the highest concentration. So it means that this solution had less water potential. So this cell lost water to the solution. So this makes sense also. Then 3, cell Z, this one, had less negative water potential than the sugar solution it was put in. Less negative implies high water potential, as we know that the highest water potential is 0. So the less the negative, then the more it is close to the highest. So this also makes sense, because we can see when it was put in, they're saying Z had less negative water potential than the solution it was put in, and that's why it lost water, because it had higher water potential than the solution. So also this makes sense. So that implies 1, 2, 3, all of them were correct. Moving forward to number 18, during telophase of mitosis, a scientist stains the chromosomes of a diploid animal cell with fluorescent dye to observe the telomeres. The cell has 22 chromosomes. So we know in telophase, chromosomes have already separated. So in metaphase they align at the equator, then within the anaphase they have separated and they start moving towards the opposite end of a cell. So we can be assured that our chromosomes are separated. So each chromosome is simply having 2 telomeres. How many telomeres will be with the scientist observe in one of the nuclei. So if there are 22 chromosomes, it means that each one is having 2 telomeres, so there will be 22 times 2, which will give you 44. Number 19, methotrexate is a drug used to treat cancer. It can act as an enzyme inhibitor, preventing the synthesis of nucleotides containing thymine. Okay, cells treated with methotrexate are not able to complete the cell cycle. A cell entering telophase is treated with this drug. Which stage of the cell cycle will be affected by the action of this drug? So they're saying it prevents the synthesis of this nucleotide, and this nucleotide is crucial because we need nucleotides to carry out DNA replication. So if we don't have this enough nucleotides, then there will be no synthesis of new strands of DNA. And as we are aware, this does not happen in the anaphase or metaphase or prophase, but it happens in the interphase of the cell cycle, specifically in the S phase. Okay, moving to number 20. The question was, which stage of mitosis is correctly described? In anaphase, chromosomes line up across the equator. No, this is metaphase. In metaphase, the centrosomes replicate. No, this happens in the earlier stages, such as prophase. In prophase, chromatids move to the opposite poles. No, this is in the late stages, such as anaphase. In telophase, chromosomes uncoil to form chromatin. Yeah, this is true. As the cell is preparing for cytokinesis, they will uncoil to form chromatin again, and then they can go into a new cell. Number 21, the diagram represents a nucleotide containing cytosine. So they are saying this base is a cytosine. Which statement about this nucleotide are correct? The carbohydrate could be a ribose. Yeah, it could be a ribose. It could also be a deoxyribose. So makes sense. The organic base contains nitrogen. Yeah, all bases contain nitrogen. Base pairing occurs with guanine. Yeah, we know that this cytosine pairs with guanine, and then thymine pairs with adenine. So, okay, it makes sense also. Cytosine is a purine. Not really. Purines involve only adenine and guanine. Then pyrimidines involve thymine, cytosine, and then uracil for RNA. So this doesn't make any sense. So we can eliminate all answers which are having a 4, and so on A 1, 2, 3, which is correct. Okay, then moving to number 22. A section of DNA contains 73 bases. So in total, we are having 73 base pairs. Yeah, it's important. Base pairs. Then they are saying number of bases on each strand: adenine, cytosine, guanine, thymine. Then strand 2. Those are the bases. So according to the Chagaf's rule, we know that in a given DNA segment, adenine number, the number of adenine bases should always be equal to thymine for DNA, because they pair together. So if we have 10, even here we should be having 10. And then on the other hand, for the guanine should always be equal to cytosine. Okay, so it means that if we have 29 adenine on one strand, so this should be pairing with exactly 29 on the other strand of thymine. So this can give us a clue that thymine should be 29. Why? Because thymine pairs with adenine on a different strand. So if we check through the options, I think they make it a little bit simple, because we can see that Z is 29 and nowhere else that we're having 29, so most likely A makes sense. But maybe for the purposes of learning, if we are to complete this question, so if we have 21 adenine on one strand, then on the opposite strand we must be having also 21 thymine. So it means that W should be also 21, because thymine pairs with adenine on a different strand. Then what else can we conclude from here? If we have 14 guanine on one strand, then we must also be having 14 cytosines on the opposite strand, because they are pairing together. So far we have confirmed this, this, and then also this. But then when we check on this, when we check on this strand, let's say maybe strand 2, remember they have said we have 73 base pairs, so it means that 21 plus 14 plus y plus 29 should be giving us a total of 73. So if we are to do mathematics, we shall find that y is exactly 9, because this will be 21 plus 14, that is 21 plus, 21, then 29, 21 plus 29, that is 50 plus 14, that is 64, then plus y is equal to 73, so your y will be equal to 9. So if we have guanine as 9, then we should be having also 9 on the opposite strand as for cytosine, because they are pairing together. So here the best option was A. So we just need to remember the pairing rules of these bases. Number 23, the photograph shows a section of a stem which labeled part is a xylem. Okay, so we know that xylem is this will be the xylem. So the best answer here was C, as for the xylem. That was a little bit easier. The number 24, 24, some fungi cause wilting in crop plants by growing within the xylem vessel elements. Which process will be directly affected by these fungi? Okay, it is affecting the xylem vessel, and we know xylem vessels is for transporting water molecules and mineral ions. So cohesion between water molecules, they're talking about water. Development of root pressure, not really. Mass flow during movement of assimilants, this is for phloem. Uptake of water by root hair cells, no. So the cohesion of water molecules within the xylem vessels will be affected. So the best answer was A. 25. Which features have the role in the transport of water in the xylem vessel elements? Capillary action, yes. Adhesion and cohesion. Hydrogen bonding for cohesion, again yes. So here the best answer was all of them, 1, 2, 3. So the best answer would be A. Okay, moving to number 27. The statement describes events that occur during the movement of sucrose. So these are the statements, and then they are saying which sequence correctly describes what happens in a plant at the source during movement of sucrose. So at the source, so the statements we are having: hydrostatic pressure decreased, hydrostatic pressure increased, sucrose is loaded. Okay, those are the statements. So when we check on the options, then we can try to verify. On A, they are saying the first one is 3, that is sucrose is loaded into the phloem vessel. Okay, so we know once this happens, what follows? Usually the water potential within the vessel will be lowered, and then this will be followed by water moving in, and then hydrostatic pressure, and do that. So let's check and see what happens next. They are saying 7, the water potential decreases, and that is true. Then 5, water moves by osmosis into the phloem all the way from the xylem, again that is also true. And then this pressure will be causing the hydrostatic pressure to increase. So A seems to be more probable. Let's go and check these other options. That is B. 3, the same thing. Sucrose is loaded, and then they are saying jumping to 8, water potential increases in the phloem. No, it's actually the opposite. So this is out. And then C, 4, sucrose is loaded from the phloem vessel. It is the other way around. Sucrose is loaded into the phloem vessel, not from. So that makes C and D unlike answers. So we go for A, 3, 7, 5, and 2. Moving to 28, they are saying which are present in the walls of the capillaries. So we know the capillaries are having the squamous epithelium that makes up this endothelium, and there are no smooth muscles, no elastic fibers, and no collagen fibers. So which option gives us that? It's only A. There's endothelium, but the rest are all absent, and that makes sense. So 28, the answer was A. 29, the photomicrograph shows 3 white blood cells labeled X, Y, and Z. A quick one, we can see this is a monocyte. So we have a monocyte, and then this is a neutrophil, and then this is a lymphocyte, and then these are red blood cells. Which row correctly identifies these cells? X, we already know it is it's a monocyte. Okay, then Y being a neutrophil, then Z being a lymphocyte. I think this was an easy question, so the answer would be C. Moving to 30, which sequence of raters correctly identifies the order of events during the cardiac cycle? So we have these events. We need to just remember the cardiac cycle in its sequence. So if we are to check the options, A, V, where is V? V is here. Wave excitation enters the atrial ventricular node. The best way to answer this question is to look for the first step in all this, and then we try to follow it up. So the first step, of course, we have the natural pacemaker, which is the sinoatrial node. So wave excitation spreads from the sinoatrial node, and then what we know that this spreads to the atrial walls, and they will be contracting. So this should be our second step. And then this excitation continues to the AVN, so this should be our third step. And then we know what AVN does. This one delays the impulse for about 0.1 seconds, so this will be 4. And then this continues down the pancreas tissue, so this will be 5. And eventually the ventricles will contract from bottom to top, so this will be number 6. So if we can check the options which are having these sequences, starting with number 1, which is X. So X, like if you check on X, then T, number 2, V, number 3, U, number 4, W, number 5, and then Y, number 6. So we can see that A makes sense. You can also cross-check all these other options. You realize that the sequence is not matching up. Okay, so moving forward to number 31, the graph shows the effect of 3 different partial pressures of carbon dioxide on the oxygen dissociation curve for human hemoglobin. So we have the partial pressure of CO2 and then also the saturation. So what do we need to remember? We need to remember that once carbon dioxide is increased, then the graph will always be shifting. But the question is, it shifts from where to where? So we know that if we have more carbon dioxide, then the affinity of hemoglobin for oxygen reduces. So now checking on the options here, what effect does this decreasing the partial pressure of carbon dioxide have on hemoglobin? Let's cross-check these options. It is less efficient at taking up oxygen and less efficient at releasing oxygen. It can't be less efficient at all events like releasing and uptake. So taking up, it is true, but on the other hand, releasing is not less efficient, actually it is increased. Then B, so A is out. Then B, it is less efficient at taking up oxygen and more efficient at releasing oxygen, decreasing the partial pressure. So here we are decreasing it and it's the opposite. So this would be the opposite. If we decrease, then it can take up more oxygen. It is more efficient at taking up oxygen. I think this is around the lungs. So it is more efficient at taking up the oxygen and less efficient at releasing it. So this is more convincing. C. It is more efficient at taking up oxygen and more efficient at releasing. Again, it can't be in one direction, it is always antagonistic. 32. What is correct about the transport of carbon dioxide by blood? 1. The enzyme carbonic anhydrase catalyzes the formation of carbonic acid in red blood cells. Yeah, that is true. To form carbonic acid. Then number 2, carbon dioxide diffuses from the active cells to the red blood cells and react with water to form that carbonic acid. So these are correct. Then 3, carbonic acid dissociates to form hydrogen ions that combine with hemoglobin to form carbaminohemoglobin. This is not true. It actually forms hemoglobinic acid. So 3 is out, but we are convinced about 1 and 2. So the answer here, which is more probable, will be B, 1 and 2 only. 33. Which statements about the all bronchioles, this is important, all bronchioles are correct? All of them having the goblet cells? Not really. The upper section of the bronchioles, they will be having some goblet cells, but as you move towards the alveoli, the goblet cells disappear. So not all of them have got the goblet cells. Cilia is present. Then muscle tissue, the smooth muscles are there for all. So here the best answer is 2 and 3. So there is 1, we can ignore all that. So we shall remain with D, which is 2 and 3. Going to 35. The diagram shows some of the pathogens that cause diseases in humans and some of the ways they are transmitted. So the pathogen can be a protocotis like that of malaria, bacterium like for TB, mobivirus. Then here we have, I think that it measles. Then here we have method of transmission: contaminated water, contaminated food, cough and sneezes. So what is the correct pathogen and method of transmission for disease of TB, tuberculosis? It's a bacterium, and method of transmission when someone coughs, and the bacteria can be spread from one person to another. So 1 and X makes more sense, so B was the correct option here. 36. The proportion of the local population who have malaria in an area is higher than the proportion in area S. Which factor causes the difference? Area A has more humid climate than S. Maybe because this is favorable for multiplication of the vector, which is the female Anopheles mosquito. Area R is nearer the equator than S. No, there's no guarantee about being near the equator and malaria. There is higher population in area R than S. Again, they are not correlating. There is less sewage treatment. I think this would be for cholera. So the most convincing answer here would be A. 37. species X is a single-celled eukaryote. Species X has been genetically modified to produce penicillin, which does not harm the cell walls of species X. So we know that penicillin can affect the peptidoglycan, which is the main type of cell wall that is found in the prokaryotes, like bacteria. But if it is a plant, it can't be affected. What may be concluded from the information: 1. The cell walls of species X are chemically different from those of bacteria. The cell walls of species X are made of peptidoglycan. No, otherwise it will also be destroyed by the penicillin being produced. The cell walls of species X are made of cellulose. There is no guarantee, we can't be sure. It can be made of any other thing, like chitin or any other thing apart from cellulose, which is for plants. But then when you come back to option 1, the cell walls of species X are chemically different from those of bacteria. That makes sense because it means that X that is producing penicillin, and we know the action of penicillin, which destroys the cell wall, won't be able to destroy species X. So since 2 and 3 are wrong, then we can go for 1 only. Okay, moving forward to number 38. The question is, when exposed to an antigen for a second time, memory cells stimulate a secondary immune response. Which correctly shows the secondary immune response? Okay, memory cells producing antibodies, that's not true. Memory cells dividing into plasma cells. Okay, memory cells dividing into plasma cells, but again we also need to form more memory cells for future. Secretes antibodies and divides into memory cells. No, they don't secrete antibodies. Antibodies are produced by the plasma cells. So here the most probable answer was C. Number 39. Monoclonal antibodies are produced for use in diagnosis or treatment of diseases. That's true. To obtain antibodies for an antigen, a mouse is injected with an antigen. Okay, that's true. Some of the events in the production of the monoclonal antibody are listed. Plasma cells are fused with cancer cells to form hybridoma. Hybridoma cells that secrete the required antibody are identified and cloned. Okay, the B lymphocytes that recognize the antigen multiply and become plasma cells. Hybridoma divide by mitosis and secrete antibodies. Plasma cells are removed from the mouse spleen. Okay, here they were saying, what is the sequence of the first 4 events in the production of the monoclonal antibody? Okay, 3 is about the B lymphocytes that recognize the antigen multiply and become plasma cells. Okay, then going to 5. 5, they are saying plasma cells are removed from the mouse spleen. That is true. And then 1, plasma cells are fused with the cancer cells or the myeloma cells to form hybridoma. Again, that's true. So it means A and B are separated by these last 2, that is 2 and 4. 2 is hybridoma that secrete the required antibody are identified and cloned. Then 4 is hybridoma divide by mitosis and secrete antibodies. So before they can secrete the required antibody identified and cloned, they need to first divide by mitosis and then secrete antibodies. So between A and B, B would be more convincing. Before hybridoma that secrete required antibody are identified and cloned, themselves they need to first divide by mitosis and then start producing antibodies, and that is how we can identify them. Okay, lastly, number 40. Which row describes passive immunity triggered by the antigen? No, that would be active. Involves the immune response? Again, no, because here you are getting or you are receiving already made antibodies. Memory cells are produced? Not really. Again, this is just for active immunity. Permanent protection? No, this is also wrong. So all of them are crosses, and from this table The one that is having crosses is D. So number 40, the answer was D. Yeah, thank you for watching. See you in another video.